Welcome to today's Explainer. We are diving straight into what is essentially the ultimate upgrade to mRNA technology. We're going to look at exactly how scientists are completely re-engineering the medical miracle that got us through the pandemic, literally tearing it down to its studs and building it back up so it's more precise, more powerful, and way more efficient.
Look, first-generation mRNA vaccines were absolutely revolutionary. No doubt about it. They totally changed the course of global health. But, just like any version 1.0 tech, they definitely have a known hardware limitation. Right now, to get a strong enough immune response, you need relatively high doses. And honestly, if you've ever had a booster, you know firsthand that higher doses mean higher reactogenicity. We're talking about those brutal side effects, fevers, chills, muscle aches, you name it. So the big question the scientific community has been obsessing over is, how do we keep that incredible protection but totally ditch the heavy side effects?
Well, the answer boils down to efficiency. The ultimate goal here is to get maximum potency out of incredibly tiny, low microgram doses, to basically eliminate those side effects altogether. But getting there isn't just about tweaking one little thing. It actually requires a simultaneous triple-threat upgrade to the vaccine's vehicle, its chemical recipe, and its internal instruction manual.
So, in today's Explainer, we're going to tear down this version 2.0 anatomy. First, a better delivery vehicle. Second, the perfect LNP recipe. Third, upgrading the instruction manual. And finally, the ultimate stress test. This roadmap from the Mitchell lab lays it all out beautifully. It targets three specific components: the ionizable lipid structure, the overall lipid nanoparticle formulation, and the untranslated regions of the mRNA itself. And the end goal, driving hyper-targeted, super-efficient delivery straight to the lymph nodes.
All right, part one, a better delivery vehicle. Okay, so let's unpack this. That fragile mRNA, it desperately needs a protective vehicle just to survive the trip into your cells. We call these ionizable lipids. Now, first-gen vaccines use lipids like SM-102, which, work fine, but researchers wanted better. So they synthesized and screened 96 brand new biodegradable options. Through all that rigorous testing, they found a stereo-defined lipid, meaning its 3D shape is meticulously engineered, called H9T6. And what makes H9T6 a real game-changer is how it reacts to pH. It acts like this super-efficient delivery truck that only becomes positively charged when it hits an acidic environment. That lets it easily break right out of the cell's endosome, which is kind of like the cell's loading dock, and just dump its mRNA payload directly into the cytosol where the magic actually happens.
Moving on to step 2, the perfect LNP recipe.
So a lipid nanoparticle isn't just one single lipid. It's a highly precise mixture. Scientists used a strategy called design of experiments to test out all these different ratios to find the absolute perfect balance. And they actually found the magic recipe. By relying on a specific helper lipid called DOPG and pairing it with super low levels of PEG, literally just 0.5%, they figured out how to radically supercharge the delivery.
And to put that into perspective, how much did it supercharge delivery? Try a massive 3.5-fold increase in getting into the dendritic cells. Those are your immune system's primary sentinels, compared to the old SM-102 benchmark we use right now. I mean, it fundamentally blew the old standard out of the water.
And when you look at this recipe inside a living system, the difference is just wild. The older SM-102 formulations kind of spread heavily throughout the body, and they really tend to pile up in the liver. But the new H9T6 formulation, it uses albumin-mediated transport, which is essentially hijacking proteins right in your bloodstream to stay laser-targeted at the injection site and the lymph nodes right nearby. That is exactly where you want a vaccine going to train your immune system, completely avoiding wasting the payload on the liver.
Which brings us to step 3, upgrading the instruction manual.
All right, so we've got the delivery truck and the perfect fuel mix. But now we got to rewrite the actual cargo. An mRNA molecule is way more than just a recipe for a viral protein. It's got these untranslated regions, or UTRs, on both ends. Think of them like the formatting on an instruction manual. The 5' UTR at the front acts like a factory foreman. It recruits ribosomes to start pumping out proteins. Meanwhile, the 3' UTR at the back controls the mRNA's half-life, so the cell doesn't just shred the instructions before the job is even finished.
Now, researchers didn't just guess what the best format for these instructions might be. They actually biologically stress-tested over 630,000, 630,000 unique 3' variants by tracking their decay alongside 68,000 5' variants to see how well they loaded ribosomes. It was just this massive, high-throughput optimization protocol to nail down the exact genetic sequences that would maximize protein production. And out of those hundreds of thousands of combinations, one undeniable winner emerged, a sequence they dubbed the 5B8 scaffold. When they really put it to the test, this specific architecture significantly outperformed the benchmark UTRs from both Pfizer and Moderna, maximizing peak and cumulative antigen expression. Honestly, it's just a far superior set of software instructions.
Finally, step 4, the ultimate stress test.
Okay, so the hardware is upgraded, the software is completely optimized. But it's one thing to see amazing results in a petri dish, right? To prove this wasn't just some cool lab trick, researchers took the optimized 5B8 mRNA, loaded it into those targeted H96 lipid nanoparticles, encoded it with the SARS-CoV-2 spike protein, and tested it in mice to see if it could actually generate real-world protective immunity.
Well, when introduced to a living immune system, version 2.0 was an absolute home run. Even at ultra-low doses, it triggered a profoundly strong, multi-layered defense. It easily bested the current clinical benchmarks, proving for sure that this design translates into actual real-world protection.
Actually, scratch that. My favorite part of all this is the division of labor they discovered. They found that the massive spike in neutralizing antibodies, the humoral response, was mostly driven by those new 5B8 software instructions. But the powerful T-cell response, which is what gives you that deep, long-lasting immunity, that required the synergy of both the new instructions and the targeted H96 lipid delivery working perfectly together.
So what is the big takeaway from this explainer? It's that by obsessively optimizing every single anatomical piece of the vaccine, the lipid, the formulation, and the genetic sequence, we don't just walk away with a slightly better COVID booster. We get an entirely new, fully proven framework for building much safer, highly potent, ultra-low dose therapeutics.
And that leaves us with a pretty mind-blowing question to end on. If scientists can now train our immune systems this effectively, using just a microscopic whisper of a dose to completely dodge those heavy side effects, what incurable disease is this tech going to conquer next? The future of personalized medicine and genetic therapies is about to get a whole lot more interesting. Thank you so much for joining me for this explainer, and as always, keep questioning what's next.